It's a great pleasure to introduce Dr. Stefan Thor, who's going to present a seminar today. Dr. Thor received his, you stop me if I get it, his degree in molecular biology and microbiology from Umeå University under the supervision of Thomas Edlund. He then went to the Salk Institute, I got the sequence right? Okay. Where he started to get involved in neuroscience and was did research in molecular neurobiology under the supervision of Dr. John Thomas. From the Salk Institute, he went to Harvard, where he was assistant professor, and then he returned to his country, where he's risen through the academic ranks at Linkoping University in Linkoping, Sweden. Oh, Linköping. Sorry. Okay, sorry. Linköping. Thank you. Correct my, my Swedish, please. It's been a and he's presently the director of the Wallenberg Center for Molecular Medicine at Linkoping University. Dr. Thor today will be speaking on the generation and degeneration of the Drosophila nervous system. Thank you.
Thank you. Thank you, Mark, and thanks for inviting me over here. It's my first time in Puerto Rico and arrived 5 days ago. Spending a bit of time out in Rincon and vacationing here, so good to see this wonderful country. Okay, so I gave it a slightly different title when I was communicating with Mark, but I figured this would be, today I would be, rather than giving you a sort of a really in-depth seminar on one particular topic in the lab, I'll sort of give you a bit of a smorgasbord, going through a couple of different things in the lab instead, so that you can see sort of little dips into different projects in the lab and give you a sort of a broader overview of the things we do in the lab. And as I don't have to go through this then, because Mark did a very good job at going over this. And as I should point out, that my experience in running research endeavors really started in 2007. For 5 years, I was head of research for a very large department at the medical school with 400 people, and were in charge of recruiting 8 or 10 principal investigators back then. And then right now I'm director for this Wallenberg Center, and we're in the process of recruiting 15 PIs into the center right now, both clinical and preclinical investigators. So I'm on the opposite side of the table all the time now. So anyhow, that's briefly about my background. And so, I mean, some of the fundamental questions in neurobiology that we're interested in the lab, I'm sure is quite familiar to you guys as well, is I'll be talking about today is first regarding how the CNS is generated, and you can of course break that down into many different topics, and the two ones that we study very intensely in the lab, my lab, is how cellular diversity is generated, how you make all the different types of neurons inside the CNS. And also, more recently, we've gotten very interested in the aspect of cell number control, because not only do we have to make blue and red cells and whatnot, but we also make them in very different numbers. So some are generated in vast numbers, and some probably only in a couple of hundred cells in the mammalian CNS. I also wanted to talk in the second part of my seminar about the degeneration of the CNS and some projects we've been doing on that aspect the last couple of years. That, of course, has to do a lot with genes and mutations involved in degeneration in humans, and how these types of genes and mutations actually affect neurons and glia to create degeneration of the CNS in the later part of life. And my long-term goal that somehow these things will meet up. They haven't yet, but hopefully they will in the future. Okay, so seminar outline will be, I'll be discussing first some of the pathways we identified that control neuronal subtype fate, and this is something I've been working with since basically 1988 when I started as a grad student. I'm going to talk about some of the pathways controlling cell number in the CNS, and this is more recent work from my lab. Then I want to talk about one example of how we've been modeling human neurodegenerative disease in the fly, and this is more recent work. And finish up with development of a novel assay system for studying neurodegeneration in the fly. And then I want to give some of my visions for the Institute of Neurobiology as far as I can glean from what I've learned so far. Okay, so a very good model system for a lot of these basic questions is, of course, the fruit fly, Drosophila melanogaster, and it's been used in so many different aspects of modern biology research now, and our work is primarily focused on embryonic development, which happens during the first 24 hours over here. And this is a very well-studied system that where we're able to really connect the dots from the earliest stages of development and to sort of late mature larva that hatches out, and we tie these gaps quite nicely now in the system. So the first part of my talk discussing pathways controlling subtype fate, I just want to give a bit more outline on nervous system development. So the fly CNS is generated in these patches of neurogenic ectoderm here. There's two on each side of the midline down here that makes the nerve cord. There's two patches up in the brain, anterior that makes the brain up here. In these patches of tissue, particular cells acquire a neural progenitor identity, which we call neuroblasts in the fly. And they'll delaminate from the ectoderm, move inward, and start to form the CNS proper. And we know from work by, I should say, in particular Gert Tech now and Chris Thor, we know soon every single neuroblast generated has a number and an identity down there. So it's an incredible system where we can go from early patterning events to knowing all the neural progenitor cells and understanding how they continue to develop. A great, vast amount of work has gone into studying early patterning in the fly, and of course the CNS is also patterned. It shows you three of the main cues that are patterning up the CNS. It shows you the segment polarity genes acting to pattern the neuroectoderm in these stripes. There's also columnar patterning genes acting to subdivide the developing CNS into these columnar stripes. And also you have more long-range anterior-posterior patterning as shown by the Hox genes here with particular... These Hox genes show some examples over here are expressed by different anterior-posterior parts of the CNS, some only in the posterior part, and this also helps to pattern the CNS. And because of these patterning cues, as you generate these neuroblasts shown in one hemisegment here, they all have acquired a unique expression pattern of different patterning genes, which sets up their identity, and they're identified also by position and size. Now, they will go on to divide and to make a unique lineage, and they do so by dividing asymmetrically with a neuroblast budding off a slightly smaller cell called a GMC, which divides twice usually to make neurons glia. And each neuroblast progresses along such a lineage, makes a small clone basically of cells. And each one of these, they make a unique clone containing unique identities of neurons and glia. So it's a fascinating system where you can go from very broad early patterning mechanisms, establishing the neuroectoderm, establishing subterritories in a grid fashion, identifying these NBs, identifying their clone, the identities of neurons and glia and the clone, etc. It provides an incredible way of linking together all this information, the flow of information over time. Apart from the spatial information, there's very important temporal information playing out, and this is something that was pioneered really by Ward Odenwald and Chris Thor over 15 years ago, when they identified the transcription factor cascade of important regulators that are playing out in the neuroblasts over time, and they do so in a sedge-way manner by regulating each other. And there's also other factors involved in this transition. And these type of factors, then, they provide a temporal information that's overlaid on top of the space information. And we played some part in also acting to further diversify or add more players to this cascade, but I won't go into the details of that today. Now, somehow then, space and time are integrated somehow into making a number of very unique cell fates. One such cell fate is shown down here, exemplified by the expression of a neuropeptide gene called NPLP1. So each one of these little blue dots is actually one cell and one neuron that expresses this neuropeptide gene. And you can see it's extremely stereotyped. It's one per hemisegment, and up here there's an extra one in thoracic areas. But somehow this slightly broader information, space and time, is integrated somehow with incredible precision and specify a number of these sort of unique cell fates. And the question is then how that comes about. And we've used these particular neuropeptide cells, again shown here in a more close-up, as important markers for addressing this, as sort of an end state handle to understand how space and time are integrated. And this shows again NPLP1, and it's co-expressed with a transcription factor called IS-absence, and you can see that these guys are coinciding. And in fact, we studied these cells quite a lot over the last almost 20 years now. And we identified a lot of different genes involved in specifying them. There's also another neuropeptide expressed right next to this one, also part of this cluster of cells over here, called FMRF-GFP, and you can see the expression of this guy here. It's only in 6 cells per in the CNS at this stage. And to find genes controlling neuropeptide specification, we've used the enhancer of this gene hooked to GFP, and we've done a large-scale screen that we recently published of about 10,000 mutant lines to identify genes important for neuropeptide expression. I won't run you through the list of that, but here's some example of these types of genes, and you can see that here's the normal expression of neuropeptide cell bodies and their axons extending here. Here are a couple of mutants that have, complete loss of these cells or partial loss of expression of neuropeptide. In all these cases, actually the cells are there, they're not lost, but I'm not going to show you data. So this is really a late specification of the cell that's going wrong. So this is the cells I talked about, the cluster out here. There's also a set of these dorsal cells. So these are all expressing this apters transcription factor, and together with IS-absent, they are important for these cells, but also provide incredibly good markers for these sets of cells, many of which are neuropeptide cells. We and others identified a number of regulatory genes and markers that can be used to highlight these cells over here. So we've done a lot of work focusing on this particular group of cells out here, which is this cluster of cells laterally, which expresses the NPLP1 neuropeptide gene, as well as the FMRF neuropeptide gene. This is called the apters cluster. One of the more important things that we've been doing is this kind of data here shows you that these genes are important for specifying these cells. This doesn't really tell you much about how they act. And to really understand this better, we have to identify the progenitor of these cells. We were quite lucky in that we managed to get hold of a marker called ladybird, which is an enhancer that we can use to light up this particular neuroblast. So this is one neuroblast and the clone of the neuroblast shown in each hemisegment. You can see the clone developing over here as you go along. And in fact, this clone, then this particular neuroblast generates these 4 apters cells shown here by the different transcription markers, transcription factor markers. And later on, you can see expression of the neuropeptides within this cluster. And so this provides us with a handle on the origin of these cells, and it provides us with a handle for looking at the effect of different mutants, what actually happens in the various mutants when you lose the neuropeptide, where the cells never generated, cells die, etc. What actually happens. It's been important for the last 10 years. And so just to summarize a lot of work that we've been doing over the last 15 years or so, almost 20 years, I guess. This is our current knowledge. So you generate this app cluster group of 4 neurons from this neuroblast in a sequential manner. initially, these 4 cells, the 2 neuropeptide cells and the other 2 cells, are all equivalent as far as the transcription factor cocktails they express. But then rapidly, in the course of a couple of hours, they differentiate into these 2 different neuropeptide cells and 2 other types of interneurons in between. The way this comes about is in a very complex manner, and this is sort of a scary cascade that you have to look at it too carefully, I guess. But anyhow, it tells you how this comes about. And here are the different neuropeptide cells. Here's the MTLP1 neuropeptide cell. Here are 2 other interneurons. Here's the FMRF-GFP neuropeptide cell. And what happens is that the whole set of upstream activators, these types of temporal genes, together with spatial information, so space and time, are integrated to activate these types of feed-forward cascades. And there's 2 different types of cascades playing out to create proper transcription factor cocktails in the final cell over here. And this is also comes about by the action of a set of repressors in the middle that are activated by the same cues. And these guys play out a little battle in there, and then you end up with 3 different cell types. And I know it's kind of a complicated slide, but this basic concept: feed-forward cascades, upstream activators, and then a set of repressors in the middle playing this out. And you can see the potency of these guys. So any of these genes, when mutated, will lose neuropeptide expression, but they have very different roles. But if you only look at the end marker, they will all look the same, loss of neuropeptide expression, but they actually do very different things. You can see the potency of these factors. I should say also that all of these factors are selective in their expression. None of them are expressed by all progenitors, by all neurons. They're all selected in one way or another, either in space, time, or highly selected in only some sets of neurons. Now, when you misexpress then certain upstream activators, like shown here, ladybird or collie, you can activate a lot of these aptrous types of cells and lots of more neuropeptides. And if you co-express them together, you can get dramatic effects, and you get a lot of these transcription factors coming on, the late guys, and lots of neuropeptide expression coming on. And you can in fact even see that the axons are reprogrammed. So a lot of the ectopic axons are now following this common axon bundle over here. Now, if you take the terminal cocktails, instead you can get even more massive expressions. And this is a terminal transcription factor cocktail activating MTLP1. And here's a terminal cocktail activating FMRF-GFP. You can see that you get dramatic activation of the neuropeptide by these cocktails. And strikingly, if you switch one of the factors, you exchange one transcription factor here to a different one down here, you can completely switch the activation. So in the same CNS as this one, it has no activation of FMRF-GFP. And this CNS, which is the same CNS as here, has no ectopic activation of MTLP1 by switching one factor. So the cocktails are highly potent, but they're also highly selective. And what these guys are doing here is just make sure that only this particular cocktail is expressed in this cell, only these cocktails come on over here. Okay, very complicated, but you get the basic message, which is that we have decoded for these cells coming knowing nothing 20 years ago, just having neuropeptide cells, starting to put transcription factors in there, finally identifying the progenitor, and being able to dissect the whole pathway of how you make these cells. Now we can make them at will in many other types of neurons. Now, and for the second part, we have to deal with how you control cell numbers. Again, you have these progenitors in here, neuroblasts. They make the clones. And they do so by dividing in an asymmetric manner, as I pointed out. The NB divides to make a daughter cell called a ganglion mother cell, which divides once to make 2 neurons glia. And this division mode didn't even have a name, because that's how we thought it was always playing out on the fly. It was given the name type 1 by Chris Doe and colleagues in 2008. And the idea was that since daughters divide once, you would call this type 1s. And the reason was that we were learning that there were actually other division modes coming along. So it's actually got a name finally. Now, we've been using a number of different markers for lineages. I showed you the ladybird marker for 5, 6 over here before we can use other markers to pick up other clones, other lineages, and study them in detail with a number of different markers. What we learned from that is that, in fact, the NB56 that makes these aptrous neurons I was talking about, it actually has a peculiar switch. It starts out in the type 1 mode, But the last 5 cells are born directly from the neuroblast. So the neuroblast buds off a daughter cell that immediately differentiates into a neuron without an intermediate daughter in there. And we refer to this as type 0 now because daughters don't divide. Chris Doe actually identified this a number of years ago for a different lineage. And we've been studying more lineages, and in fact here's a very nice lineage. It's called NB33A. It doesn't matter what it's called, but that's what it's called. This shows you very elegantly how this plays out. So down here is the stem cell, the neuroblast down here, and the white stain is the dividing cell, phosphorhistone 3. Now you can see the neuroblast divides throughout development. At every point you can see about 20–30% of cases the neuroblast is dividing. Early on you have daughter cells up here that are dividing as well at a high frequency. But very rapidly there's no division of daughters, and for a long period of stages there are never any dividing daughter cells. And we've done this in cell death mutants, etc., so we can verify this is not a cell death effect. And it basically tells you this one has a very short type 1 window and has a long type 0 window. It's very nice lineage for looking at effects of type 1 to 0 problems. So there are 3 of these types of lineages. They all start as type 1, switch to type 0. So is this something that's prevalent for the whole CNS then? Now we can use markers to globally look at neuroblasts versus daughters. So neuroblasts can be identified by asymmetric staining of Prospero, nuclear stain of deadpan, and then phosphorhistone 3 for division. And daughters then have nuclear Prospero, no deadpan. So these can be distinguished quite easily, which means you can look at that globally in developing CNS and distinguish dividing daughters from dividing mothers on a global scale, up and down the whole CNS. And what we see is that early on, there is a roughly 100% match. So it means that every dividing neuroblast seen as a whole group has an equivalent dividing daughter cell. But rapidly during development, this drops down to much lower percentages, which tells you that what we saw for 3 specific lineages is probably true for the whole CNS. But many neuroblasts, perhaps all of them, undergo this particular type of program change. They start making daughters that divide once, finish up with making daughters that directly differentiate into neurons. Now, we've been trying to identify which are the key cell cycle genes that control these switches, which are the key upstream regulators that control these switches. And I won't run you through all the data, but this is one elegant example, I think, of such a gene. Again, here's the wild type scenario, the dividing neuroblast, the mother cell, early daughters, and then no divisions of daughters up here with a white stain. Here's a da capo mutant. This is a fly equivalent of P21 KIP and P27 KIP, cell cycle inhibitors. You can see in these mutants that the daughters never drop out. So you have a very high percentage of daughters dividing throughout, and the embryo core keeps dividing. And this tells you that these mutants have a complete failure to control this switch. They stay in the type 1 mode throughout development. And of course, this makes a larger lineage. We can also quantify that. Now, using these types of single lineages as markers and the whole global analysis, we can be able to look at more cell cycle genes and a lot of upstream regulators. And I'll just summarize that on one slide. Now I should show you also globally that the capo also has a global effect. So it doesn't look so striking. You see some more division, but if you quantify daughters versus mother, you can see that there's a complete failure to drop down in these mutants. So you stay 100% matching mothers to daughters throughout development as opposed to dropping down in the wild type control. So we think they all start out as this, and they all, most perhaps all drop down into type 0 over time. And then the molecular cues are as such. Work from several other people, in particular Harvey Racine and Andrea Brand, has shown that this particular transcription factor called Prospero, which plays a very important role to control this particular proliferation mode. Now, Prospero is a transcription factor, but it's cytoplasmic in the neuroblasts. Goes asymmetrically to daughters, enters the nucleus, where it starts to downregulate these 3 very critical cell cycle genes. That means that this daughter can divide once, but never again. So you make 2 neurons of glia. And of course, since Prospero never goes into the nucleus of the neuroblast, this guy can go on and on and on forever. But what happens then over time is that the temporal gene cascade plays out, that I talked about before. You hit castor, which is a key player here, and you start to up-activate Hox genes, and you start to activate the notch pathway in here. And these together will activate expression now of the capo, which is not expressed over here. Notch activates enhances split downstream genes, and this suppresses these same transcription, these same cell cycle genes up here. This creates the switch then. So now the daughter is not allowed to divide at all, and this is highly controlled, but can undergo multiple rounds of this because this protein is actually degraded in the neuroblast over here. So this is a different mechanism for making repetitive type zeros over here. Eventually, castor activates the final temporal gene grain here, which will then repress even further cyclin E and E2F, which creates a stop of the neuroblast over here. It's another important decision to stop the neuroblast on time. And that's where we are right now, but we're continuing to identify more regulatory genes up here that can control this. So what's striking with this is the precision of the decision. It's not as if these guys go down sloppily type 1 and suddenly just get tired and start making type 0 daughters. This is happening at very precise time points, but it can be different for each lineage. It can happen very early or late in the lineage. So somehow the neuroblast, the progenitor identity with the space will be integrated into this type of information. And then you will stop, make the switch at a certain time point, and you will stop at a certain time point. This creates a very precise lineage size in neurobiology. But there's lots more to do in here. Now, why is this even interesting from a broader perspective? Well, as I told you now, we have identified in the field of the people type 1, and we have found type 1 to 0 switches in here. But again, the reason this name came about is because actually at that time point, 3 different labs identified what's called type 2 neuroblasts. And these are very large neuroblasts that make very large clones up in the brain. There's 8 of these per brain lobe in the larva of CNS. And these make lineages of 500 cells, something like that. So they're very large lineages. And the reason is because they have an amplification step. So the neuroblast makes a daughter cell that in turn divides 5 to 6 times and makes QCs and then 2 neurons. And it goes around multiple of these rounds. And that's, of course, a way of amplifying the system tremendously to make a lot of these cells. So this is the system in Drosophila. But why this is even more exciting is because if you look then at mammals, studies of, for instance, mammalian cortex have identified that neuroprogenitors, in this case called radioglia cells, can generate neurons directly, can generate daughter cells that divide once to make 2 neurons, or can generate transit amplifying of basal progenitor cells that can generate daughters that divide multiple times. And of course, we would call these then type 0, type 1, type 2, although in the mammalian field, that's not what they call them. They have actually many names, actually too many names, I would say. It's actually confusing a bit. But the basic 3 cassettes are there. These are evolutionary conserved cassettes. The question is then, are the switches in here stereotyped and programmed, or are they random and filling up niches? And of course, are these switches linked to the same cell fate? Because what hopefully became clear from the first part of my talk was that concomitant with the change from type 1 to 0, you also start to make different cell types. So you switch proliferation mode, you switch the type of neuron glia you make. That controls the number also now. So numbers and fate are integrated right away in that aspect. Now, why this is even more exciting, I would say, is because there's a huge interest right now in the mammalian field understanding the wrinkleness of the brain. Or the gyranocephalic brain, as you know, and of course, lower, or I don't know if you should use the word lower, but I mean, for instance, rodents have a smooth brain, a so-called lissencephalic brain. And as you start to move up the animal ladder into cats and humans, you start to get a very wrinkled brain. And it's widely believed, although it's very difficult to prove, that the wrinkleness is important for smartness. Because you actually, by wrinkling things up, you'll be able to connect certain dots much more efficiently. It's very hard to prove or disprove that idea, actually, but there's a lot of literature supporting that notion. Now, the most now this morphogenesis that happens in here and here compared to here could be controlled by many mechanisms, of course, folding and things like that. But one widely held hypothesis right now is that it's controlled by daughter proliferation. So that up here you would have an expansion of type 2 behavior. Again, it's not called type 2 mammals, but it's the same kind of thing. So that here you have a lot of these type 2 behavior. And down here in rodents, this is extremely scarce. And in fact, I haven't seen any paper actually describing a clear type 2 proliferation mode in the rodent right now. But mostly you're talking about what we call type 1 is 0. But up in primates, you start to see these type of amplifying cells. So it's believed that progenitors, by having more proliferative daughters, will expand the outer surface of the cortex. That starts to wrinkle it up, and that makes you cleverer. So it's a very big deal, actually. It may not be completely true. It could be controlled by morphogenesis, cell cell adhesion, or whatever. But
how does the fly fit into that?
How smart are the flies? That's a good question. Well, I should point one thing out. That first of all, we also think that there's a very clear anterior-posterior gradient to this. Because even though you find these guys up here, you don't find them down in the spinal cord, even in higher mammals. And if you look at the fly then, these 8 cells are only found up in the anterior-most part of the brain. And down here, we're all down to type 1 and 0. And in fact, we have a paper about the route soon. We show it's actually a gradient of type 1 to 0. So there's more type 0 back here in the fly. So we think there's an evolutionary ladder from type 0, 1, 2. I think there's an anterior-posterior gradient from type 0, 1, 2, which has to do with expanding the brain and wrinkling it up eventually. So it's a very big deal how you control these daughters. Other than it's very fascinating how precise it can be fly in a certain lineage, but that's more esoteric, but it has an actual impact. And in fact, there's a number of human disorders, micromegacephalic disorders that are believed to be linked to daughter proliferation problems. So there is some sort of medical importance to this, and there's some people with less or fewer wrinkles, etc. But it's probably not just the daughter proliferation that makes it. It's probably very precise morphogenesis, because you actually form these folds, the fissures are formed in very precise places and in very precise shapes and patterns. So it's not enough just to make a lot of crazy daughters dividing to make it wrinkle. Then it become randomly wrinkled. It probably wouldn't make you very smart. So it's precise linking. Okay, so the third part, modeling human degenerative disease in the fly. This is something that's been done by many labs over the last 15 years. It's pioneered really by Nancy Bonini and Mel Feeney's lab, in the first 2 pioneers, and one brought us also to say. And we've been sort of following in those footsteps a bit, but trying to improve some of the assay systems. So the one disease we've been focusing on is AD, of course, and as you know, this is a very severe and prevalent disease in Western societies, and highlighted by these plaques containing a lot of amyloid, and of course these sort of tangles inside neurons containing a lot of tau. And we've been focusing a bit on APP and the generation of amyloid and A-beta. So APP is a large molecule that's cleaved, and it processes out a small fragment called A-beta that can be quite toxic, but this comes in very different flavors, and this is complex processing cartoon, but I'm sure you're familiar with these things from many other talks and papers. But there's been a lot of issues around the A-beta peptide and how it actually works and how toxic, why and how it's toxic. And there's a lot of ideas about aggregation of A-beta peptide and what actually makes, what's the most toxic thing for neurons. Is it these types of tetramers or other types of tetramers, and what goes on? It's also very complex because due to different types of cleavage and also post-cleavage degradation, you generate a lot of different types of peptides, not just 142, but also other peptides. And so we wanted to try to understand systematically the toxicity of these different peptides in the fly system. And now we're looking at adult CNS, of course, of the fly for these experiments, which is quite a lot more complicated than the embryo, but still orders of magnitude less complicated than rodents or humans. So this for misexpressing A-beta peptide with the GAL4-US system. So you have flies expressing GAL4, transcription factor from yeast, and you have your responder, which is upstream activating element driving your favorite gene of interest, in this case A-beta, and this will then target expression to certain tissues. So we've been developing the Galafor side of things, improving that. One of the problems was that this particular driver used in the system, in a vast majority of papers, has a robust expression in adult brain. This is showing GFP now, but it goes down quite a lot over time. So even though it's very broad and it's very robust, it's not very good for looking at adult long-term effects of driving various types of toxic proteins. So we developed multi-transgene inserts of this particular transgene instead, and this now has robust expression over the course of several weeks into the adult. We've also improved the expression of the A-beta peptide by using a better signal peptide, a better consensus start codon, and codon optimizing the ORF to get expression higher. So with that, we made a series of A-beta constructs to express all known variants of the A-beta peptide in the fly. And one benefit, and one of the reasons we wanted to do this, was that in the fly there was the development 5-10 years ago, sort of landing site transgenesis. So one problem we have when you're trying to do structure function in a transgenic system is that most systems give random integration of your construct. So you have your DNA plasmid, you make mutations in it, you put it into a mouse or a fish or a fly, and it goes anywhere. And of course, depending on where the transgenic insert lands, it's going to be affected by the environment in that area, by the epigenetics. And so now you start to compare really apples and oranges. You mutated one amino acid, but the thing is in two different parts of the genome. And then, of course, the expression levels can be completely different. So really, it's problematic. That's been a big problem in the field for worms, for flies, for mice, everything. Now, the landing site transgenesis, you have a landing site engineered first by a transgene, which has an at P site in it. So it means that you can land your favorite plasmid in the same exact base pair all the time. So there's an inbred genetic background, you have a landing site waiting there, and all your plasmids go into one of the same base pair. Now you can really compare structure function much more systematically. That's a huge advantage, I think, for this field now. So we find what many other people find. We find that A-beta 142 peptide is extremely toxic, and in our system it's more toxic than anyone has published before, because we improved the system both on the Galafor side and the US side. So now the flies are super sick, and average lifespans are down to around 10 days. We can also try various other types of deletion. We find that the 342 peptide is also extremely toxic. We also find that 1142 peptide is extremely toxic. So these are three very toxic variants of A-beta. This is partly known before. I should give credit to the whole field of it. This is known from other systems, but this is the first time we can test it systematically in vivo in a landing site setup. So that's important, I think. And we can test all sorts of other variants of this. And we also been looking at ELISA using the Mesozoic scale, which electrochemical ELISA, very sensitive. So you can look at soluble and insoluble, and you can of course find that there's tons of insoluble A-beta 142, whereas the other peptides are more soluble. And this goes hand in hand with toxicity, of course. And same for 342 peptide is also highly insoluble, high fraction insoluble A-beta 342. We also used a novel type of staining tool developed at our university, which is a polymer-based staining. This is a synthetic polymer that has fluorescent capacity. But depending on how it interacts with proteins in the tissue, you actually emit different spectral signal. So this is a staining of a human brain. You can see it stains the plaques in one spectrum and it stains tangles in a different spectrum. So it can be used to actually distinguish in a section different types of aggregated proteins. Now, if you looked at the control fly, early on there's no real staining in some weak background. Over time, you start to pick up a little bit of staining in adult, in late flies. Sorry, slightly mature fly. That's probably because you start to pick up some misfolded protein in the system itself over time. Now, if you're looking at the non-toxic peptide variants, there's really no massive pickup of state signal over time. Of course, if you look at these very toxic ones, there's massive buildup of these aggregates that stain very intensely with the polymer. Then this tells you that there are indeed protein aggregates in these brains. So to summarize this, we established a much more robust A-beta model by improving the US side, improving the Galafor line, and using landing site transgenes. This gives you very precise structure function capacity. And again, we find very high toxicity of these three peptides, and with some interesting aspect, we've been mutating these residues, but I won't go into that as well, but some interesting findings as far as the tail end of these peptides. And it emphasizes toxicity of these positions for the tail ends. We can find these aggregates using polymers, and that protein solubility goes hand in hand with toxicity. And we're doing a number of other types of studies like this to use the landing site technology to really study structure function or other types of disease proteins also in the fly. And this is something that can be readily done nowadays. I mean, we've gone from we almost don't do anything nowadays. We construct on the computer things that's being ordered with a company that synthesizes the DNA. The company sends the DNA to another company that injects the DNA and sends the flies back to us. And that's more cost effective than grad student and postdoc messing around with building DNA, injecting flies. You know, so it's a weird, and it's not that expensive, strangely enough. Okay, the last part then has to do with novel assays for the effects of these types of proteins in the fly. Of course, not only A-beta is toxic to the fly, but many other types of disease proteins are toxic to the fly. And this shows some more recent data testing sort of toxic versus non-toxic variants. Here's A-beta 140, 42. You can see it. 1 to 40 is completely non-toxic compared to control. 142 is extremely toxic then. And the same thing is true for wild-type tau versus a very toxic synthetic tau. You can see it drop down here and other types of polyglutamine expansions and parking and whatnot, other things. And in general, the wild-type version of the protein is not particularly toxic or completely non-toxic, whereas the mutant version is very toxic. You can also see that on geotaxis. This is a locomotor assay. You can see that many of these things are extremely give extreme effects on geotaxis pretty rapidly in the fly. So again, the question is, what actually happens in the neurons? I mean, we know these proteins are toxic since many years, but what actually happens inside the neurons, and how do you actually assay that? And we were intrigued a couple of years ago by studies from Matthew Freeman's lab and also Nancy Bonini using the fly wing, because of course in the wing you have sensory cells, and so there are sensory neurons out in the wing going into the brain, and they were using that to study how neurons degenerate out there by just cutting the wings off. And so we thought maybe one could use the fly leg for this. This idea wasn't lost. This idea wasn't very unique to us, and so recently actually Matthew Freeman published a paper using the leg to look at various aspects of neurodegeneration. But anyhow, we've been working with this for 3 years now. So this is the story as far as we work with. So the fly is up here. Here's the adult CNS. You have motor neurons with their cell bodies inside the CNS. They project out and have terminals over the muscles out here in the leg. There's also sensory neurons out here. The cell bodies are out here, and also some of them here. They project back into the CNS to innervate and land on different interneurons in here. This is a nice system where if you look down here, there aren't any motor neuron projections, so you can look at purely the cell bodies and dendrites of the DNS cells. And if you look over here, especially out on the side, you can look purely at the motor neuron endings on the muscles over here. Now, so we've been going through a number of fluorescent markers available in the field, and some of our own markers also, to look at how robust they are. If you look at the adult leg and look at what they actually label. These are different types of fusion proteins with RFP, Ruby, GFP, etc. And some of them were quite interesting and informative. So it shows you some examples. Here you look at mitochondria GFP. This has been used by other students. It's not very exciting, but you can see the mitochondria in the nerve endings over here. You see red is labeling the whole nerve. So you can see the mitochondria nicely spaced out. Here's an endosome marker, LAMP1 GFP, has different RAB molecules, nuclear GFP to see the nuclei over here, etc. Well, so these are different markers, but you can use these then, you combine them with the Galfo line, and you can drive different disease proteins and see what actually happens to the different disease proteins. Is there anything interesting to learn about what the disease proteins do? How do they kill the fly, actually? How do they kill the cell? And starting with something very, very, very simple, you can use nuclear GFP to simply count and look at the cell death. So these are the different nuclei of the sensory neurons out here. in the TARS, and you can see that you can count the nuclei quite nicely, and it's very it's a very small standard deviation. In fact, the deviation is probably mostly that sometimes because of the cuticle, it's a bit difficult to scan through the cuticle with confocal. Sometimes you don't see the cells or what, but there's some variability, but it's quite reproducible. Now, at day one, even with these really nasty disease proteins I just showed you, none of them, apart from one motor protein, shows any loss of cells over here. In contrast, moving out to day 10, 14, now you see a menu such as A-beta 142 and poly-Q expansion of Huntington has a profound effect on the numbers of cells, and you see a severe loss of these cells. So this is a very straightforward way of just assaying cell death, and it's quite rapid. A different example is this nice molecule, Lifeact-Ruby. This is a synthetic molecule from yeast fused to ruby red that stains F-actin. And this is then the PNS cell bodies out here with the dendrites and the axons going to the CNS. And you can see Lifeact-Ruby stains this F-actin network right close to the cell body very nicely. Now, if you look at some of these nasty disease proteins like A-beta, you see a real mess in here. Because this is partly because cells are dying, but you can still see the cell bodies, but it's real mess with F-actin. Fluid is really bad. Here we don't have so much cell death at this stage, but you see real mess. And tau, the dominant tau molecule, has a real profound effect on Lifeact. And you can quantify this more carefully, and this shows you just a quantification where you can see long F-actin bundles, shorter ones, and very short ones, and sometimes even the control slightly fragmented F-actin bundles, and different types of other aspects. You can see that early on at day one of a newborn, there's not that many super long F-actin bundles, but as you go into later on, you see very nice long F-actin bundles going up to about 56%, and very few of these short fragmented things. Now, if you then look at the disease proteins, it's a real mixed and interesting bag. So some things like blue then completely wipes out, and you're mostly left with fragmented F-actin bundles, but also ataxin has a very profound effect, giving a lot of fragmentation. Other things such as SCA3 has a lot of these fragmented F-actin bundles, fragments out in the axon cells, not close to the cell, but out in the axon, so you get an expansion of those types of stuff. And anyhow, lots of these kind of interesting features in here that happens with this marker. And so the idea is that by using lifespan geotaxis and a growing list of these types of markers that will tell you about different aspects of the neuron's life and its homeostasis, you can start to sort of barcode the disease proteins efficiently, see what they actually do, what kind of aspects of a neuron's life are they affecting somehow. And this is something we're pursuing with force. Okay, so I should acknowledge people before I move to the final part. So, as part of the cell fate, it was mostly conducted by Magnus and Daniel, grad students in lab, and more recently by Johannes Stratman, Shas, Parampur. This was also pioneered in the lab many years ago by Irene and Doug, who are now running all labs around the world. It's been a collaboration also with Jonathan Benitez-Siepel's lab in Madrid. Second part, cell number story, was also Magnus and Daniel, but with important inputs from Karina and Caroline here. Also Ryan and some others have been involved in this. Part of the notch work has been done with François Schweizkut and Khalil Massouni in Paris. And then the modeling disease part is done in collaboration with Per Hammarström, who's a great protein chemist and a grad student in his lab. Also a very talented postdoc in the lab, Margosata. Annika is a great technician. The leg neuron model was done by mainly Josephine Fernius, who's a postdoc. Again, Annika and Helene and Caroline are excellent technicians in the lab to work with the leg project. Okay, final part before we finally have a lunch break, I hope. So visions for the Institute of Neurobiology. It's obviously not very easy to this being my first visit here, to have a great grand vision for the whole institute. But I think the first thing, if I was to be offered this position here, would be sort of course to listen and learn, to see what people are interested in doing here, and what would be a good way to move the institute forward. And then, of course, discuss this with the whole faculty, co-workers, and also, of course, the Central University, form a strategic plan for the next 5 years, and then to act upon that. So sort of a no-brainer, I guess, in that sense. What I would say is that what I know from being both in the San Diego area, Salk UCSD, and also in the Harvard area is that basic biomedical research is extremely competitive, both in the US and globally, of course. So, I mean, it's at the risk of offending people in the audience, it's unlikely that an institute like this will sort of be able to take on these biomonsters head on and develop research avenues that are directly competing with some of these big institutes. Rather, one should sort of build on unique potential that this has, being sort of an interesting maritime and tropical area, which is unique to the US and unique in the world almost, I would say. There are not many places that do cutting-edge research in this type of research. That's why I think this center that was launched here several years ago to sort of look at environmental neuroscience is a very smart initiative, because there are obviously lots of unique things happening here with respect to environmental change that you know, you can't really recreate somewhere else. I think also these initiatives are also very important and make a lot of sense to me coming from the outside. Other areas that I was thinking about is sort of in the area of natural pharmaceuticals, because you have a wide variety of very complicated and unique species, both on land and in the ocean here. And I learned from Emma this morning that there is activity at the main campus in these areas. This is something that could be interesting to develop as well. And of course, being a developmental biologist at heart, I would say that the area of evo devo is also very interesting. Of course, again, you have access to a wide variety of different species, right, essentially outside the doorstep, basically. And of course, you know, any initiative will depend upon common goals and also, of course, funding, because initiatives usually cost some money. But I would say that I think the geographical and cultural location of UPR at the intersection in Latin America, the Caribbean, and the US presents a unique opportunity with tremendous potential for growth. And that there's several different strategic initiatives launched. This new recruitment ongoing of junior faculty, and some junior faculty also joined fairly recently. And so I think it's a possibility that we could see some substantial development here over the next 5-10 years or so. Yeah. Okay, with that, I should thank you for your attention. It's my kids having fun at Rinkoping a couple of days ago.